Hello everyone, and welcome to today's live broadcast, IHC Protocol Optimization and Troubleshooting Tips, presented by Thomas Ruyle, a scientific support specialist at ABcam. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by ABcam. ABcam is an innovator in reagents and tools. ABcam's purpose is to serve life sciences researchers globally to achieve their mission faster, providing the research and clinical communities with tools and scientific support. ABcam offers highly validated biological binders and assays to address important targets in critical biological pathways. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time during the presentation. Just click on the green Q&A button located on the lower left of your presentation window and type your questions into the box that appear on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, notice that you're viewing this in a slide window. To enlarge that window, just click on the screen icon located on the lower right. If you have any trouble hearing or seeing this presentation, click on the support button at the top right or use that Q&A button and let us know you're having a problem. I'd now like to introduce our speaker, Thomas Ruyle. Welcome, Tom.
Okay, I think we're ready to go. Hi everybody, this is Tom, and welcome to the webinar. This presentation is a really simple kind of introductory talk about IHC protocol optimization. If you've been doing IHC all along, you may not have anything new to learn from this, but if you are new, hopefully this will help you out. And if you have any questions, of course, you can email them to us down the road, and you'll have some contact information at the end of the talk for that. Okay, let me use this. Okay, sorry about that. Moving along, so the agenda is going to be basically how to choose an antibody and IHC optimization. And we'll start off with how to choose an antibody. And this is assuming you don't have one already. You're searching around looking for an antibody to use in your work. And what a lot of people do when they're starting out, they'll just go straight to the Google and they'll type in some search terms. And the ones I typed in here as an example are CD31 antibody, CD31 being the cell surface protein, and then IHC to qualify the search so that you're looking for an antibody that's useful for CD31 IHC. And you'll get a list of antibodies, basically these first few hits, and the results are ads paid for by various antibody companies. As you scroll down, you'll see other vendors. And one thing you can do, it's kind of useful when you're doing this kind of really broad search, is just to click on Google Images, and that'll bring up lots of images of different antibody staining for CD31. And if you click on each individual image, it'll bring you typically to the vendor page. Some of these might be coming from publications. But a better way, a more focused way to go about looking for an antibody would be to go to specific search sites that specialize in antibodies or bio reagents in general. And a few examples that I listed here, they are going to be collecting antibodies from multiple sources, academic as well as commercial, and you can filter the search results by tested applications, species reactivity, host species, and other criteria. So it's a little more useful than just simply doing a Google search or any kind of a broad search engine. Examples are CiteAb, Linscott's Directory, BioCompare, Antibodypedia. There's a bunch of them. But a lot of these sources will ask, and some ask, you must ask to actually be listed with these sources. Sometimes you have to pay to be included in these databases if you're a vendor selling antibodies. So that won't necessarily get every antibody out there. just the caveat, something to be aware of if you do use those resources. So if we're even more focused, if you just go straight to a supplier's website, and I've chosen ABcam conveniently in this case, and I started to do an example search here for a protein called P38, which is a kinase. And this search, just typing in P38 on the website search box brings up 21 results. And those can be filtered by applying different kinds of filters. These are listed on the left-hand side in this example. And you can filter by species reactivity or application. And after applying some of those filters to this list, it came down with 2. I filtered on antibodies that have been knockout validated. And since I've done this, actually, this has returned 2 results. Since I did this a few months ago, we've actually added many more antibodies that have been validated against knockout samples and comparing them to wild type. And this is something I'll discuss in a moment, this kind of validation. But now we have these 2 antibodies that have, as a result of this search, and the top one is a rabbit monoclonal antibody, I believe, and the bottom one is a mouse monoclonal. And we're just going to pick that one at the top there because it has a nice image of immunohistochemistry. And this conveniently has a caption which describes the protocol that was used to produce this image. And so this is normal human esophagus, formalin fixed, paraffin embedded. And you see this staining pattern, which is consistent with what's expected for P38, a nuclear stain with some light cytoplasmic staining. This is a protein that will shuttle back and forth between the nucleus and the cytoplasm. So this is basically what's expected. And this caption is useful because it tells you exactly what was done to achieve this particular stain. Just a 15-minute incubation you'll see too, which is kind of short. These kinds of protocol steps can be modified to a certain extent depending on what's convenient. The incubation time in particular, and also the detection method. This was detected with a secondary antibody conjugated to horseradish peroxidase, so it gives, and then the substrate gives you this brown stain. And the blue is a counter stain with hematoxylin, just to give you some orientation as to where things in the tissue are. Part of the validation of this particular antibody was against the knockout sample. So this is a Western blot showing you 4 lanes in addition to the molecular weight markers on the far left. And the first lane is a wild type sample, and the stain for P38 is shown up in the green here. And then the next lane over is the same cell line, but with the P38 knocked out. And you see there's no green there in the blot. So this is basically just telling us that the antibody is in fact detecting P38. And the last 2 lanes are just HeLa cells and Purkat cell lysates. Showing then, this is basically also showing us the specificity of the antibody. There's no cross reaction with other proteins, which would be apparent above and below. The red is the stain for, I believe, GAP-TH, which would be a loading control, just to demonstrate that there was even loading of protein across all 4 lanes. And so now we've covered how to choose an antibody, we're going to move on to IHC optimization. And this is a pretty busy little slide here showing the basic protocol that one would use to stain a tissue section, and specifically a paraffin embedded, a section of paraffin embedded tissue or frozen tissue. You're starting off with the fixation step first of all for the sample, and then you're moving on to embedding and sectioning, and those first two steps are critical. The choice of fixative is critical insofar as some antibodies are not going to be able to stain any tissue that was fixed with a formaldehyde-based fixative, and and some aren't compatible with that kind of fixation at all, so you would have to either use frozen tissue or just move on to a different antibody if you're locked into using or staining paraffin-embedded formalin-fixed tissue. And then the sectioning itself, there's some variation there. You can cut a thin section, which is the most common kind of sample to be stained, 5 to 10 microns thick. If you're working in a neuroscience lab, you might be sectioning much thicker things like 50 microns or so and using a free-floating technique. And I'll discuss that a little farther on, I think. And then the next step after that, after you've sectioned your slides, or sectioned the tissue that is, and placed it on a slide, the antigen retrieval step serves to reverse some of the effects of formaldehyde-based fixation. And for most antibodies, this is necessary, or at least if it's not necessary, it will improve the intensity of the stain if you simply apply this antigen retrieval step. And that can be a heat-induced retrieval, which entails actually boiling the section on the slide, or an enzymatic retrieval with enzymes like proteinase K or pepsin or trypsin. And then to point out that a lot of people wonder if they can actually apply a heat-mediated retrieval step to a section of frozen tissue. If the answer is yes, frozen tissue tends to be a little more fragile than a section that's been fixed with formalin and embedded in paraffin. So the remedy would be to use a slightly lower temperature, maybe 90 degrees centigrade or 80 degrees centigrade for a longer amount of time, instead of just boiling the section for 10 to 15 minutes, which is the standard protocol for a paraffin-embedded section. That's pretty harsh. Like I said, then with a frozen tissue section, especially if you're working with skin or certain other kinds of tissue, boiling is just going to knock it right off the slide, and you'll ruin the morphology at the very least. So lower temperature, longer incubation time can remedy that. The next step at number 4 here would be blocking with the non-specific binding of the antibody. So you want to use a percentage solution of serum from the secondary antibody host species, and then once you've blocked all this possibility, ideally, of the primary antibody sticking non-specifically to proteins in general, you move on to step 5, which is actually incubation with the primary antibody. And then the optimal concentration and incubation, that's typically going to be obtained from the data sheet of the antibody. In the cases where the antibody has never been tested for immunohistochemistry before, you'll need to do some experiments to determine what the optimal dilution of the antibody is. Incubation can range from 15 minutes, like we saw earlier with that P38 antibody, or you can go up to, you can just incubate overnight at 4 degrees in the refrigerator. The main point to remember is never to let the section dry out. Once it dries out, you're going to end up with lots and lots of non-specific binding of the antibody. You'll see just no specific staining. No specific staining that can be distinguished from all this background staining. So keep everything wet all the time. Don't let anything dry out if you're incubating it overnight. And way to prevent things from drying out if you're incubating overnight is to keep them in a chamber with, say, a wet paper towel, just a humidified chamber, and then place it in the refrigerator. Then the next step would be, after washing off excess primary antibody, incubation with a secondary antibody, and then finally rinsing off the excess of that and incubating with the substrate if you're using an enzyme for the detection, or no substrate if you're simply using a fluorescent detection method, fluorescently conjugated secondary. Okay, so we're going on that. It's a very, very standard protocol. And the poor results generally of 3 kinds. You'll see no staining at all, or you might see lots of staining, but it's this kind of general background, non-specific staining, with or without specific staining. So you might see some staining that looks correct, but it's kind of overwhelmed by all this kind of background, non-specific staining. Third kind of problem would be seeing some staining that looks correct or specific, but is incorrect. I should say not doesn't look correct, but it looks like there's certain things within the tissue that are staining specifically and intensely, but it's incorrect staining. It's, say, staining for a protein where you should exclusively see it in the nucleus. You might see lots of staining of the cytoplasm, but no nuclear staining, or a protein that's supposed to be mitochondrial. You might see just nuclear staining. So that would be an example of an incorrect staining. Or incorrect cell types. You might see staining of a cell type that is not expected to express the protein at all. And the causes of these poor results can be broadly classified as either sample issues, protocol issues, or reagent issues. And let's start with no staining due to sample issues. And one of the main causes would just simply be that there's no protein to detect. The protein expression is low or absent. And so you want to be sure you're looking, your expectations are valid. So you want to review the literature. And this is an example. Let's see, this is a protein expressed in mouse, in mouse tissue specifically, that is found in macrophages, but it's absent from some macrophages which are localized within T-cell areas of lymph nodes and spleen. So very specific expression of this particular protein. So you should be sure, like I said, to look for it where you would expect to see it, which isn't to say that you might discover something completely new, but you would have to do lots more experimentation to confirm that. And that same protein in human is expression is restricted to eosinophils. So this, what I'm looking at here, this database record on the right-hand side is from Uniprot, which is a very useful tool for looking at where proteins are expressed. It has lots of literature references to go along with the sections in this database record that refer to tissue-specific expression. And another resource we like to go to is called the Human Protein Atlas. So if you're looking for, in particular, a positive control, some kind of tissue where people have reported detection of this protein through a variety of different assays, Western blotting, ELISA, or what have you, and immunohistochemistry, this database, Human Protein Atlas, will have lots of suggestions. It presents tissue RNA expression data and also tissue microarray IHC data, but their tissue microarray data for many proteins is from just 1 or 2 antibodies. So their ambition is to collect lots of data from lots of different antibodies against any one given protein. And so definitely look at proteinatlas.org, and you'll see lots of examples of these images from the tissue microarrays. By tissue microarray, we mean a slide that contains multiple tissue types, all stained with the one antibody, and it's a very convenient way of looking at where something is found in various tissues. And see, moving on to no staining due to sample issue, poor sample preparation can be a factor. So over or under fixation of the tissue or insufficient removal of paraffin could lead to a negative stain. And it can be difficult to diagnose poor sample preparation, but if multiple antibodies against several different targets fail, it needs to be considered. So tissue samples prefixation should be no more than several millimeters in thickness. I think I wrote diameter here, but what we mean is thickness, so that the fixative can permeate the entire tissue. Formaldehyde-based immersion fixation, for instance, with paraformaldehyde or formalin, that should last for at least a minimum of 24 hours. Perfusion fixation, which is another method of fixing the tissue, that's going to typically be shorter and followed by a post-fixation, post-immersion fixation. Overfixed samples can be salvaged more easily than underfixed samples. By salvaged, I mean the overfixation can be reversed with antigen retrieval. that boiling that I mentioned earlier, the antigen retrieval, that will reverse overfixation. Underfixation is a little more difficult, so you have to remember that the fixative is moving from the outside of the tissue to the interior. If it's underfixed, the interior will have no fixation, and then subsequent steps in the procedure might denature those proteins in the interior, and you'll see just differences in staining intensity across the tissue. No staining due to reagent or protocol issue. So these are a little obvious, but we'll go over them anyway. If the antibody concentration is too low, you want to use a series of dilutions, including the dilution recommended obviously on the data sheet. That recommendation on the data sheet might hold for the way that the antibody was validated, but your tissue is going to be different. It might be fixed for a longer amount of time, might have less of the protein, so you might see, even if you get a negative result, that just simply using more antibody will produce the positive stain, or at least will intensify a relatively weak stain. With a secondary antibody, you want to check the data sheet for isotype specificity and species reactivity. So, for instance, an anti-rabbit immunoglobulin secondary antibody may not be able to stain a mouse primary antibody. So you want to match up the species reactivity of the secondary with the species of the primary antibody, and also the isotype. Some anti-mouse immunoglobulin secondary antibodies are isotype specific. So isotypes would be IgG1, IgG2A, 2B, IgG3, and so on. And one way to check would be just to check against another primary antibody, the secondary antibody is, check that against another primary antibody to ensure quality. If it fails with another primary antibody or multiple primary antibodies, there's a question of the quality of that secondary antibody. No staining due to reagent or protocol issue. Moving on to antigen retrieval. I mentioned this heat-mediated retrieval at various, you can use various pHs. Citrate buffer, pH 6 is really popular. And another one would be more alkaline, tris-EDTA at pH 9. And then another variety altogether of antigen retrieval would be enzymatic retrieval with trypsin, proteinase K, or pepsin. And I'm showing here some data from a paper that compared different pHs of antigen retrieval buffer, and they were examining two different antibodies. One was against progesterone receptor on the left, and another was looking at an antibody against E-cadherin on the right. And you see the results at the lower pHs, the E-cadherin antibody failed altogether. pH 3, progesterone receptor antibody stained intensely, and then also at pH 9, relatively weaker at the pHs in between. So just to show that each antibody has its own requirements for the pH of the antigen retrieval buffer. And this paper actually was much bigger. It showed this big table of lots of different antibodies they tested under lots of different conditions, and they graded the intensity of the staining with these plus marks from none at all to +3, 3 plus marks, and just again demonstrating that the antigen retrieval itself needs to be optimized for each antibody. Again, the data sheet from the vendor should have that all squared away for you, so it shouldn't be necessary for you to do it yourself. But a histology lab receiving an antibody that haven't worked before, more often than not, they'll test two or three different pHs and potentially enzymatic retrieval, which is simply digesting the tissue section with one of those enzymes I mentioned. Off-target or background staining due to sample issue. So if you're looking to use a fluorescent detection, say a directly conjugated antibody, the primary antibody directly conjugated to a fluorophore, or using a secondary antibody conjugated to the fluorophore. The issue of autofluorescence comes up a lot, especially in the green channel, and you'll see lots of red blood cells, especially that are fluorescing very bright green. Typically, you can ignore the red blood cells since they're easy to recognize as such. Another issue would be with enzymatic detection, endogenous peroxidase or phosphatase with peroxidase. You can quench that with the peroxidase with you can quench that with peroxide, or the phosphatase with levacimol. But yeah, the remedy for the autofluorescence, which I didn't mention, was to simply use a fluorophore with a different wavelength. Sometimes just moving from green to red will remedy that. Okay, off-target background staining due to sample issue. Here we go. If using an avidin biotin complex for detection, the ABC will detect endogenous biotin in the tissue. And this can be diagnosed with a negative controlled tissue slide, which does not receive any primary or secondary antibody, just the ABC reagent. So this avidin biotin complex, which has horseradish peroxidase or phosphatase attached to it. This is a detection method people use to amplify a signal. If there's endogenous biotin in the tissue, that ABC complex will stick to that, directly to that, and create all kinds of background staining basically wherever the biotin is. And so there's a way to address that, and it entails simply incubating the tissue section with some avidin, and then that avidin in turn will stick to the endogenous biotin. And then if you follow that with some biotin, since the avidin has multiple binding sites for biotin, you want to occupy all of those. So it's just simply adding this extra blocking step of avidin, then followed by biotin, in addition to the standard serum block. And that should take care of any kind of background staining due to endogenous biotin. Off-target or background staining due to reagents. So we're getting close to the end now. The blocking reagents, you want to check for contamination of the serum stock, and you might want to try a different reagent, for example, 5% bovine serum albumin, if the blocking appears to be ineffective. The primary antibody, again, if you're seeing very weak staining or very strong staining, you can test serial dilutions of that primary antibody to find the dilution that's just right. And you can increase the number of wash steps. If you simply, after the primary antibody, just wash once very quickly, that might be insufficient, and there might still be some primary antibody soaked into the tissue section that needs to be removed. And then finally, you can include some detergent in the antibody diluents and wash buffer, for example, 0.1% Triton 1X100 or tween 20, and that will help reduce the amount of non-specific staining you might get. Primary and secondary antibody controls, so these are pretty important. Isotype control in place of the primary antibody reveals presence of unblocked FC receptors on various white blood cells, for example, B cells and macrophages. And a secondary only control, no primary antibody, will reveal detection of endogenous immunoglobulin in addition to FC receptors. So the tissue itself might have some endogenous IgG that the secondary antibody sticks to, and including this control with just no primary antibody at all will it should reveal if that's the issue, if that's the problem or not. If you see just with a secondary antibody just lots and lots of background staining, then you have some issue with that. And the remedy would be simply to choose a different secondary antibody. And a lot of this, what I've gone over pretty quickly. There's some nice videos on the ABcam website. And this one, these two I picked out, sectioning of paraffin-embedded tissue video protocol. So this will actually show you the sectioning itself. You might have a histology lab that's doing all this for you, but it can be instructive to actually see exactly what they're doing. And then also we have the video for IHC of paraffin-embedded sections. So this will go over, you'll see somebody actually going through those steps we discussed earlier. And if you really run into a lot of trouble, of course, you can always contact us, and these are the contact details for ABcam. And that wraps it up. I'll bring it back to this contact page so you can look at that and copy things down if you like. But if you have any questions, you can always contact us by email or by phone. Okay.
Thank you so much, Tom, for that informative presentation. We will now start the live Q&A portion of this webinar. If you have any questions you'd like to answer, please ask now. Just click on that green Q&A button at the lower left of your presentation window, type your questions into the box that appear on the screen, and click the send button. We'll answer as many questions as we have time for. So let's take a look at our audience questions that are coming in. Our first question, Tom, is why would one want to prepare thick sections, for example, 50 microns or more, rather than thin sections?
Basically, to get more information. So if you want to cut a thin section, that's typically what people are just looking at 2 dimensions. They want to see something where things are next to each other. The thicker the section, if you have a confocal microscope and you're using immunofluorescent detection, you can kind of move up and down through that Z-plane, that thickness of the section, to see things that might be difficult to find otherwise. And so a lot of times people working in neuroscience are going to be working with thick sections of the brain or the spinal cord, because those cells have lots of processes and dendrites, and it's easier to see cell contact if you're looking at a thick section. It just gives you a better opportunity if you can scroll up and down through the Z stack with a confocal microscope to see the things that might not be obvious or might be hard to find if you're working with a thin section. So that briefly would be why you would want to work with a thick section, and people working with brain. Also, you should point out that people working with these thick sections are using a free-floating staining technique. And that's where you're actually, rather than placing the tissue section on the slide, you're just letting it float around in a bath of antibody solution. Because it's so thick, you want to ensure that the antibody is diffusing into that thick section. And one other wrinkle for that kind of protocol, the free-floating immunohistochemistry, is that you need to permeabilize with some kind of detergent, because that section is so thick it's going to contain some intact cells, and in order for the antibody to get inside of those cells, if necessary, you need to permeabilize this plasma membrane with the detergent. So that's another variation of thick versus thin, free-floating versus staining directly on the slide. But that, really briefly, was why people want to stain a thick section and how.
Thank you so much, Tom. Our second question from our audience is, which is more sensitive, fluorescent detection or colorimetric detection?
It's difficult to say. It depends. There's different varieties of colorimetric detection. So by colorimetric, we mean typically using an enzyme followed by a substrate of that enzyme, which lays down onto the tissue section this insoluble precipitate. And that can be very, very sensitive. There are ways to amplify that signal. I mentioned the avid and biotin complex earlier. And you can also use something called tyramide signal amplification, which is even more, it gives you even more sensitivity. So if you're using those kinds of extra tertiary steps, the third steps, you can get extremely sensitive. Fluorescent detection as well as sensitive. People are using that mainly just because it gives you this greater contrast. If you're trying to stain more than one protein, you have the option of staining in the green channel versus the red channel, and then maybe a counter stain for the nuclei in the blue channel. So the fluorescent detecting can also be very fluorescent. It can also be very sensitive, but it comes down to choosing a good primary antibody more often than not. So it's hard to say. Long story short, it's hard to say which is more sensitive, but I think in general, people would say colorimetric detection with the proper tertiary technique would be more sensitive.
Thank you so much, Tom. And I want to thank our audience for their participation today. Our last question is, What are the advantages of frozen versus paraffin-embedded tissue IHC?
Third question. What are the advantages of frozen versus paraffin-embedded tissue IHC? The advantage of frozen, well, one of the big advantages of frozen is that it's frozen tissue is that it's minimally processed. So you can imagine a tissue section that's going through paraffin processing. That's removing all the water. In order to remove all the water and replace it with molten paraffin, you're placing the tissue in a solvent that's compatible with molten paraffin. So it starts off with ethanol, varying degrees of percentages of ethanol. And then that's followed by xylene, and then eventually you have this molten paraffin at 60 degrees centigrade. So that's a lot of treatment of the tissue, and in a lot of cases that kind of these solvents and this high temperature paraffin can destroy the epitopes that an antibody wants to detect, and simply snap freezing a tissue instead is much less harsh on the tissue. The worst that could happen might be is kind of, if you're trying to store them long term, kind of a freeze-thaw cycle might eventually destroy the epitope of the protein. Briefly, frozen's less harsh, and another big advantage of frozen versus paraffin-embedded tissue would be that the speed. It's much faster. In fact, a lot of during surgery, there's a type of surgery, microsurgery, called Mohs, M-O-H-S. Which was developed by Frederick Mohs in the 1930s. And this is actually what somebody's having a skin tumor treated or removed. The surgeon needs to know exactly how much tumor has been removed, and to the point, and not removing too much normal tissue. So they'll actually take this tumor off the skin and then freeze it and do an IHC while this patient is still on the table and come back with the results, and they might have to do that again. So you can get actually all this information within an hour. You're doing this frozen immunohistochemistry, whereas paraffin processing takes, more often than not, the whole process just goes overnight. It's going to take at least 5 to 6 hours to get the tissue from fresh to embedded in paraffin. So that's the big advantage of frozen. It's very quick. The advantage of paraffin-embedded IHC is that typically the morphology is preserved better. There's more detail in a tissue section that's formalin-fixed paraffin-embedded compared to a frozen tissue. So that's in a nutshell.
Thank you so much, Thomas. I'd like to thank you again for your wonderful presentation. I'd also like to thank LabRoots and ABcam for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through April 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Bye-bye.